You're the one doing that turnaround. What are you doing, Al? Hold on, hold on.
What's up? Oh, dang, I cursed you. I'm sorry. Dang. I wouldn't wish that on anybody. Soggy body.
It's not like that. It's not like soft.
Oh, but it's just soft. Yeah. You might get a soft bottom. I don't know if it's worth it.
Okay, let's get started.
There's nowhere in the syllabus for that, right?
Let's get started. So last Monday, you guys learned substance transport in the kidney. Today, I am going to continue talking about how substance moves through the plasma membrane. And my focus is going to be in the brain instead of the kidney. So movement of substance into and out of cells happen all the time, constantly. This process is vital to life because it's not only the cells need to. This is not only this process. It's not only for the cells to acquire what they need and get rid of what they don't need. It is also important for cell communication and a proper cell function. So to give you an example, neurons in the central nervous system, in order for them to properly function, they need to control what substance stay inside the cell, what substances stay outside the cell. So in other words, the plasma membrane of the cell assists in the maintenance of the homeostasis of the cell by maintaining a balanced equilibrium between inside and outside environment of the cell. Okay, so many different types of substances cross membranes through different mechanisms. So this is the focus of my lecture today. As I said, all the substances need to pass through the plasma membrane in and out. So let's start by talking a little bit about the plasma membrane. So the plasma membrane is the boundary that separates the interior of the cell from the surrounding environment. So the plasma membrane is primarily composed of phospholipids, which conform so-called lipid bilayer. See, this is one layer on top, this is one layer, this is extracellular, this is intracellular. So this is, we call the lipid bilayer, which is due to the phospholipids stacking. This phospholipid bilayer forms this plasma membrane, and many substances can cross the plasma membrane, like ions, water, proteins, small proteins, and molecules. However, the plasma membrane has selectivity for these molecules or ions. It allows some substances crossed over more easily than the others. So what are the factors that dictate the permeability of the plasma membrane? Size, what else? Charge. Why is it charged? Are there any charge in here? Are this charged? Okay, what else? Correct. Okay. So as I said, phospholipids are the primary component of the plasma membrane. The phospholipids has hydrophilic head on both sides, and they're exposed to aqueous solution. Phospholipids also has hydrophobic tails. They line to each other. So one tail of one side, the tail of one side of phospholipids line up with the tail of the other side of phospholipids that cause the formation of the bilayer layer. This, here's some, a detailed structure of this. You can see they're all packed together with the bilayer layer. In addition to the phospholipids, there are other components in the plasma membrane. One of them is cholesterol. You can see they are interspersed across the plasma membrane. What does cholesterol do to the plasma membrane? Increase what? The what?
Fluidity.
Well, actually, the cholesterol is increased rigidity of the plasma membrane. They're interspersed. We're going to talk about in the next slides also. In addition to the cholesterol, there are two types of protein you can find in the plasma membrane. One is called integral membrane proteins. So this type of proteins, they are spread across the plasma membrane. And the other type is called a peripheral membrane protein. This type of protein, they stuck on one side of the plasma membrane. They do not span across the plasma membrane. So do you know any examples for integral membrane proteins? No. Have you guys learned GPCR already?
Yes.
So the G protein, GPCR, typical structure of GPCR are 7 transmembrane protein, right? They span across 7 times of the plasma membrane, so they are integrated membrane protein. Transporters has 12 transmembrane. They span across the plasma membrane 12 times. Vesicular monoamine transporters, 12 transmembrane protein also, they span across. So what are the peripheral membrane protein you know so far? Remember, they do not span across, they only attach. They may attach constitutively, or they may attach to it by recruiting. So what are the proteins that are attached to the plasma membrane? G protein. So the G protein has a G alpha, beta, gamma subunit. G alpha and gamma subunit has this tail that are attached to the plasma membrane protein. So they are not integral membrane protein. They are peripheral membrane protein. What else? Protein kinases. They don't span across. They have a hydrophobic tail that can attach to anchor it to the plasma membrane so they can be interacting with the receptors or transporters, phosphorylate them, and trigger the signal transduction. Okay? In addition to, the other thing I want to comment on this is that, like you said, the plasma membrane actually has fluidity and rigidity. And the reason the plasma membrane is fluid is due to the feature of the phospholipid protein. So the phospholipid protein, the phospholipids in the plasma membrane can move within the layer, most likely bilaterally. And a lot of proteins that are embedded in the plasma membrane can also drift a little bit. It is rare that the proteins can flip-flop across the plasma membrane. This is rare, and this is more common. They can drift a little bit laterally. So if the plasma membrane has a lot of unsaturated fatty acid, then the plasma membrane is very fluid. Okay? If the plasma membrane is composed of saturated, a lot of saturated fatty acid, it's rigid compared to unsaturated carbohydrates. So cholesterol here, you can see they're interspersed across the plasma membrane. So when the phospholipids started to move laterally, the cholesterol can add a stop to it. So it has a certain degree of fluidity, but it also adds rigidity so it doesn't free flow. Does it make sense? So my question for you is why we do need the plasma membrane, the fluidity, some degree of fluidity and rigidity. Why do we need that? Why can't you have a plasma membrane that is so rigid, so every protein is properly positioned in there and doesn't move?
Can't respond to your environment then.
Be specific. What do you mean?
You have to be able to interact and
Correct.
adapt to change.
So when a stimulus comes, let's say the agonist binds to the receptor or transporters, the receptor and transporter have to go under conformational change. Some channel will open, some channel will close, right? And in the meantime, some adapter proteins have to be recruited to be adjacent to the transporter receptors, right? So for the protein kinases, G protein that have to be recruited to be adjacent to the receptor and transporters, they need a space. They need fluidity so the protein can be recruited to the targeted protein, right? So we need the rigidity and fluidity for most proteins to be functional, especially signal molecules. There are two types of molecules that can cross the plasma membrane. One is a membrane permeable. In this category, they're water, ethanol, gases like oxygen, CO2. They can free moving through the plasma membrane. There are also molecules that are membrane impermeable, such as glucose, ions, charged polar molecules like the MMS, like dopamine, serotonin, norepinephrine, and nucleotides. So this need mediators for them to cross the plasma membrane. So membrane, there are three types of membrane transport: passive transport, active transport, and a vesicular transport. Actually, vesicular transport is part of the active transport, but it's unique. It has a unique feature that is different from active transport. I understand you guys already gone through this a little bit in the last lecture. So for the passive transport, there are two types. One is a diffusion, the other is a facilitated diffusion. So for the diffusion, usually the substance brought down the concentration gradient. Okay, so you can just diffuse through the concentration gradient. However, for some substances that are big, they cannot penetrate the plasma membrane, they need facilitation. So there are two types of proteins that are embedded in the plasma membrane that can facilitate this type of passive transport. So one type is the channel proteins, like epiphorin. This is a membrane embedded protein that facilitates water transport. And this is a mechanism for the cell to maintain the water content. Okay? The second type is called carrier proteins, such as ligand-gated channels. They are also embedded, membrane-embedded proteins. When the stimulus comes, the channel opens, the molecule gets in, and the channel closes, spit out the molecule inside of the cell. Okay, so even for this type of transport, it is the molecule still go down the concentration gradient, even it is mediated by protein. So here are the examples, two types of the protein that are facilitating this diffusion. So the first one is the channel protein we talked about, epiphorin. So when water comes in, when it opens, the water can just go through this channel and it get dissipated intracellularly. And here we have the carrier protein. When stimulus comes, it causes conformational change, so the gate open, you have molecule binds into this carrier protein, and then the channel close on the top, open at the bottom, and dissipate the molecule into intracellularly. So this is a carrier protein mediated diffusion. So facilitated diffusion does not require energy, even if it's protein mediated. It's, as I said, it goes down the concentration gradient. For some transport proteins, some transport proteins, they need to move the substance against their concentration gradient. So that requires energy. Most commonly, they are adenosine triphosphate, ATP. So the active transport is performed by specific protein that embedded in the membrane and allows cells to maintain concentration gradient that different from their surroundings, means they are going to go through against the concentration gradient. The sodium potassium pump is a typical example of an active transport system. So here we have extracellular, this is a plasma membrane with extracellular space facing outward and intracellular space cytoplasma inward. So you have sodium concentration that is high extracellularly and low intracellularly. For the potassium concentration, it's high intracellularly, but it's low extracellularly. So when stimulus come, the sodium binds to this transport protein, and the ATP hydrolyzes to ADP, and phosphate provides energy, so it phosphorates this protein, causes a conformational change. Okay, then the sodium moved out of this channel protein, this transport protein, and the potassium comes in, bind to the protein. Then the phosphate is removed, conformational change, potassium kicks out of the protein and into the intracellular space. So the sodium is moved against the concentration gradient, the potassium is moved against the concentration gradient. This, every single step requires energy of ATP. There are 4 types of active transporter. So we talk about the sodium potassium ATP pump. Then there's a light-driven pump. We're not going to talk about this today. Then you have Symporter. When what does it mean is that when the molecule is moved across the plasma membrane, and so does some ions like sodium chloride. Okay. Here you have antiporter. So when the molecule moves, protons moves in opposite directions. So for a lot of vesicular monoamine transporters, they are antiporters. For a lot of monoamine plasma membrane transporters, they are C-porters. We're going to talk about it later on when we go through every single one. So I'm going to focus on mostly the monoamine transporter. So we're going to talk a little bit about dopamine transporter, serotonin or epinephrine transporter, and acetylcholine, GABA, and glutamate. So first we need to know where are the transporters located, and where are the neurotransmitter located. So here's the synapse of dopamine. So this is the presynaptic terminal from the cell body, and this is the postsynaptic membrane. So you have a dopamine that is synthesized and packaged into the vesicle. Okay, so every single, the dot here are vesicles that are dopamines. And when stimulus comes, the vesicles dock to the plasma membrane and fuse to it and release the dopamine from the vesicle. This is called exocytosis. Okay? So when dopamines are released start to stimulate the postsynaptic receptors, right? So for this reaction to be terminated, there are transporters, in this case dopamine transporters, that are located on the plasma, the presynaptic plasma membrane. Okay, so what it does is can reuptake of dopamine from synapse back into the nerve terminal. Okay, once the dopamine is taken up into the cytosol, then it can be packaged back into the vesicle for recycling. Okay, so here we have a plasma membrane that is located on the dopamine transporter that is located on the plasma membrane here. Then we have vesicular monoamine transporter that is located on the plasma membrane of the vesicles. So when the dopamine get packaged inside of the vesicle, the proton comes out for exchange. And here is a dopamine transporter on the plasma membrane. So you have dopamine comes in and sodium chloride all go in. And here you have the proton exchange with the dopamine. Okay? So same thing with the serotonin synapse. So you have the serotonin is packaged into the vesicles here, and when stimuli come in, the vesicle fuse to the plasma membrane and release the serotonin in the synapse. Again, you have the serotonin transporter seated on the plasma membrane here that can reuptake the serotonin back into the nerve terminal, and then the serotonin get packaged back into the vesicles through this vesicular monoamine transporter, which is antiporter, and this is symporter. Are you following me? Okay. Same thing with the cholinergic system. So you have acetylcholine that is synthesized from choline and acetyl-CoA. Okay, you have choline and acetyl-CoA that for acetylcholine synthesis. So when the vesicles undergoes an exocytosis, you have acetylcholine in the synapse, but acetylcholine is rapidly broken down into a choline. So here you have the choline transporter seated on the membrane, taking up the choline back into the vesicle, and then you have this antiporter vesicular transporter that can package the choline back into the vesicle and through in exchange for the proton gradient. Here's the GABA system, same thing. You have the GABA transporter here, so basically this is the channel, GABA goes through the channel, get transported in, but you do have the vesicular transporter here, packaging GABA back into the vesicles. So all the major neurotrans system works like this. You have the presynaptic terminal that is enriched in the vesicles containing the specific neurotransmitter. And when the stimulus come, usually, when the membrane is depolarized, then the vesicle undergoes exocytosis and release the neurotransmitter in the synapse. And same thing with the glutamate system. The difference between all of the system is that the ions that are utilized for the transport is different, and also for the vesicular transporter, the same thing. The gradient difference are different, like how much proton that is needed for the transporter to be neurotransmitter to be packaged in the vesicle are different. So for the neurotransmitter transporters, the plasma membrane transporter can be grouped into two categories. One is the sodium chloride dependent transporter, we call it SLC6 family, and we have high affinity glutamate transporters, we call it SLC1 family. Structurally, they're a little bit different, and the ions required are different. For the intracellular transporters, there are vesicular monoamine transporters, VMAT, VMAT 1 and 2. There's a vesicular inhibitory amino acid transporter, we're not going to talk about it today, and also vesicular glutamate transporter. So I'm going to focus on, for the plasma membrane transporter, we're going to focus on SLC6. For the intracellular transporter, we're going to focus on vesicular monoamine transporters. Here are, I can't see anything here. Here are the details of each family. For the SLC gene family, there are dopamine, which includes dopamine transporter, serotonin transporter, norepinephrine transporter, GABA transporter, glycine transporter, glycine T1, T2. There are two types. The ions that are required for this class are sodium and chloride. And there are 12 transmembrane intero proteins. And there are evidence that they are dimers, but I don't buy that. Because there's no physical evidence, there's no crystal structure showing that they are functional. They have to be dimers to be functional. I do see a few publications on the dopamine transporter. They may be dimers, they may be tetramers. So, but one thing I can tell you is that the transporter doesn't function as a monomer. They could be heterodimer, from a dimer with a transporter, or from a dimer with another receptors. But usually they don't function as a monomer. Yes?
For the SERT, does the potassium work as a counter transport in addition to the same port of the sodium chloride? Or?
So for the SERT, there's a couple of publications, maybe I don't know whether that paper talked about it. So when the sodium and chloride go in along with the substrate of the transporter, there's report that the potassium also, right. So, yeah, so I mean, even the DAT, SERT, and NAT, they are structurally similar, but there are some subtle changes in that. For the SLC gene family, the glutamate transporter, you have EA, T1, EA, T2. Some are in the neurons, some are in the glial cells. They require sodium and protons, calcium, potassium. They have, for this category, they have 6 transmembrane, 6 plasma membrane domain. Their C-terminus has 2 hairpin loop. They said it's a trimer. So again, there's no proof for that. For the, I'm sure when somebody is going to talk about this later on in your lecture. So for the weak glue transporter, this is another gene family. They have many variants. So they have they differ in the how many plasma membrane domains. So between 6 and 12. And this one here at VMAT 1 and 2, we're going to touch a little bit in the end. So this, as I said, this is antiporter. So when the substrate is transported through the vesicular monoamine transporter into the vesicles, the proton comes out to maintain the homeostasis of the cell. So here's the family for the SLC. You can see it's the largest family pathway are very, very similar. They share like between 40 to 50% sequence homology, especially the transmembrane domain, TM1 to TM12, they are very homologous. You have the GABA with different GAT1, taurine, creatine, GAT2, 3, and you have glycine transporter. I don't see leucine transporter here. It should be here. So here is the difference between these three families: monoamine transporter, GABA, and glycine. So I want you to focus on this here. For the SERT, DAT, and NAT, as I said, when the substrate goes through the plasma, get transported through the plasma membrane, the sodium ion goes in, chloride goes in, and for the SERT, potassium goes out. For DAT, it's known based on the crystal. Well, there's no crystal structure for dopamine transporter, SERT and NAT, but 10 years ago, there's a crystal structure on leucine, bacterial leucine transporter, which shares about 40 to 60% sequence homology. So based on that crystal structure, there are two sodium ions that bind to the transporter. So people believe that for dopamine transporter, there are two sodium ions that are binding to the transporter. For the SERT, maybe one or maybe two. I see publication in both cases. NAT, nobody has really studied that, but regardless how many sodium needed, all three of them need a sodium ion to go through and chloride ion to go through in order for the transport of the neurotransmitter of their substrate. For GAT, 2 or 3 sodium ion that needed, and maybe 1 or 2 chloride, depends on which one we're talking about. And here for the glycine transporter, 2 or 3 sodium ion, 1 chloride ions that are needed. So why the transport need ions to transport the substrate? To maintain electrochemical and neurochemical gradient, correct. Okay, so here's a typical structure of the monoamine transporters. It has a 12 transmembrane domain with DAT, NAT, and a SERT, okay? And it has intracellular, the N terminus is intracellular, the C terminus is also intracellular. This is very different from the GPCR you guys have learned. But the GPCR has a 7 transmembrane domain. The N terminal is what? Extracellular, the C terminal is intracellular, okay? And for monoamine transporter, there's a huge second loop, extracellular loop, and it has this glycosylation side. What does glycosylation do to the transporter receptors? Say it again.
Targets it to the membrane.
Targets the membrane. Okay. Anybody else? Okay. So if the transporter, once the transporter is synthesized in the nucleus, it gets exported from the nucleus to the cytosol and to the plasma membrane, right? If it is a mature protein, it is modified by glycosylation. If it's not a mature protein, you don't have any glycosylation. Most of the time, the protein are immature, they stay in the cytosol of the nucleus, and eventually they undergo degradation pathway instead of trafficking back to be functional on the plasma membrane. Make sense? Second point is for dopamine transporter, especially in this case, this glycosylation is involved in the ligand binding. So if you mutate the residue here, so the protein is not glycosylated. But if you put a trafficking signal, make it traffic to the plasma membrane, it is still not functional. Does it make sense? So if you remove the glycosylation side, they don't traffic to the surface. But if you add a signal to the transporter that does not have a glycosylation side, force them to go to the surface, they are not functional. So the glycosylation, we call this is the post-translational modification, is the key for the stability and efficiency of the transporter. Again, this is sodium chloride dependent. The other thing I want to point out is the N-terminal of the transporter. This is the side, usually for a lot of adapter proteins to bind. I'll give you an example. Here are the side for the binding side of the dopamine D2 receptors. They can form heterodimers. This is where they bind. And also this is the binding site for a lot of protein kinases like PKC. They can phosphorate transporter, then change the function of the transporter and cause abnormal trafficking of the transporter. So the N-terminal is very important. If you truncate the C-terminal, it doesn't alter dopamine transporter function much to some degree, but if you truncate the N-terminal, it's not functional at all. Okay? So this is proposed crystal structure of the dopamine transporter based on, as I said, 10 years ago, based on the leucine transporter crystal structure. So in that model, they proposed that the transmembrane domain 1 and 6 are key for ligand binding. And the TM3 and 8 also are involved in the ligand binding. Okay, so you have this ligand binding pocket right here, and you have the sodium binding side to sodium binding side in here. In addition to this 4 transmembrane domain that are involved in the ligand binding, the extracellular loop, ELC3 and EL, I guess 1, are also involved in ligand binding. Okay, so you can see the 2 sodium binding side, and this is the binding pocket for the ligands, and here are the crystal structure. Here's a detailed residues that are involved in ligand binding. So here's arginine 85. That's the key. If you mutated this residue, you will have a significant reduction in binding affinity of the ligand. You have this 2 sodium binding side here, and here this is another residue, alanine 314, that is involved in the ligand binding. Here again, the glycosylation side, if you mutate this 3 residue, they do not form proper conformation. Okay, have N and C terminals. So, what is function of the dopamine transporter? So we talked about it a little bit before that. One of the transporter function is to reuptake the dopamine in the synapse back to the vesicles, so the dopamine can be repackaged back and recycled for next stimulus. So here's a diagram to show you that the dopamine transporter is sitting on the plasma membrane, and when the dopamine binds to the transporter, dopamine is the substrate of the transporter. Okay, when it binds, you have 2 sodium goes in and 1 chloride go in, and then the dopamine get transported from outside into inside and get released, right? So once it's released, it can be transported to the vesicles by vesicular morning amine transporter in exchange for proton gradient, and this can be recycled and do another round of exocytosis. Okay. Okay, so this is called this process is called a reuptake. It's to terminate the dopamine activity, to terminate dopamine action that is released by either electric stimulus or by any drugs like cocaine and amphetamine. Okay, so this is called a reuptake function. It's to take up dopamine in the synapse back into the vesicle. The dopamine transporter, all the monoamine transporter are bidirectional. So in addition to take up the substrate back into the vesicle, it can also release the substrate from intracellular space to extracellular space. So we have here the vesicles that stores the dopamine, and if you give a drug like amphetamine, what it does is it causes the dopamine in the vesicle to be released into the cytosol instead of this whole vesicle docking on the membrane and undergoes exocytosis, right? So here is when you give amphetamine, the dopamine in the vesicle get released into the cytosol, into the cytoplasma, and then the dopamine transporter here the dopamine can bind to the transporter inward and then get transported outside. So we call this a process efflux, which is opposite of reuptake. Okay, so this function is very important. Why? Because if the dopamine doesn't get transported out, if it stays here, if you have too much dopamine that is released in cytosol, what it does is, and it doesn't get packaged back into the vesicle, what it does is it gets degraded right away. So one of the mechanisms, one of the hypothesized theory for Parkinson's disease is that the VMAT is dysfunctional. It dissipated too much dopamine in the cytosol and gets degraded. It can be oxidized. So this is one of the mechanisms for hypothesized mechanism for Parkinson's disease. So just remember, this is a bidirectional. It's a two-way process. You get dopamine taken up back into the cell. You also get, the transporter can dissipate the dopamine from intracellular to extracellular space. Okay? But this process is triggered by a certain substrate, not every single molecule setup has binding affinity for the dopamine transporter. So as I said, amphetamine is an example here, but if you give cocaine, which is inhibitor, it doesn't cause this efflux process. Amphetamine does. That's the uniqueness of the amphetamine. Okay, so let's talk about cocaine. Cocaine is a dopamine transporter blocker. What I mean blocker is once they bind the transporter, it doesn't get translocated intracellularly. It just stays on the plasma membrane until it dissociates from the dopamine transporter. Yes?
I'm sorry, on the previous slide, just to clarify, you said amphetamines, they cause the
Efflux of the dopamine?
Yeah, which causes them to degrade.
No. What I'm saying is that if the dopamine is released from the vesicle and stay in the cytosol, they're going to be oxidized quickly, so you have depletion of the dopamine, which is the case for Parkinson's disease. Okay, so naturally, if the cell is functional, when you have more dopamine that accumulated in the cytosol, usually this reverse transport process takes place right away.
Okay.
So does the efflux happen without stimulation?
Good question. Actually, I don't know. I got asked many times. I don't think this is a primary mechanism at a resting state, but if you, let's say, if you have food, or if you are excited about something, this may kick in as a balance act of, the reuptake. So I don't know that whether this is a physiologically happening. I don't know because nobody can test it. but I know this is a condition for a lot of drugs that can be triggered by a lot of drugs.
So my question was, when the dopamine's in the cytosol, it gets degraded, but if it goes through efflux and goes outside of the synapse, it doesn't get degraded, or?
So once it get into the cytosol, once it get into the synaptic cleft, then the reuptake process
Oh, okay, or is it?
takes over right away. But you do have dopamine diffusion here, right? Because you can't take up every single molecule that is released. So you do have a little bit diffusion here. Once they diffuse away from transporter, they're going to be oxidized right away. So if you guys work with the dopamine, you can take a little bit of dopamine, dissolve in water. If you don't add any protease inhibitor, phospholipid inhibitor, you can just watch, like after 10 minutes it turns yellow. So it's an oxidation process. So for cocaine, as I said, it's a blocker. It binds to the dopamine transporter. It doesn't get transported inside of the cell. So what happens is when cocaine binds to dopamine transporter, it blocks the reuptake process. So what happens is you have so many dopamine that are accumulating in the synapse cannot be taken up through the dopamine transporter back into the nerve terminal, right? So, and this going to be degraded. So chronic cocaine abuser, they have less dopamine content because you are basically blocking this process. And once there are so many accumulated here, they start to diffuse. Then you have more degradation going on. Make sense? And so this enhanced dopamine concentration in the synapse is associated with the reward property of cocaine. Okay? So let's talk about amphetamine. Amphetamine is not it's a substrate. When I say that, when I say substrate, it means when it binds to the dopamine transporter, the amphetamine molecule gets transported inside of the vesicle, which is different from the blocker. It sits on the membrane, blocking, okay? So amphetamine first bind, then get translocated. So in that regard, amphetamine is both a blocker and a substrate. Make sense? Okay, so when amphetamine gets in, it acts as a false neurotransmitter. So what it does, the vesicular monoamine transporter consider amphetamine as a dopamine. So it transports amphetamine into the vesicle and depletes dopamine from the vesicle. Okay, so you have more dopamine in the cytoplasma, right? Then the reverse transport of dopamine occurs. So you have more dopamine dissipate, reverse transport through a transporter into the synapse cleft. So which cause accumulation of the dopamine in the synapse. So this is a different mechanism compared to the cocaine. So cocaine mechanism, cocaine causes the increase of dopamine is due to the blockade of dopamine uptake. Amphetamine induced increase of dopamine is due to the enhancement of dopamine efflux through dopamine transporter. Does it make sense? Make sense? Okay? That's the difference between substrate and a blocker. A lot of people have studied dopamine, the dopamine transporter uptake function. So it's well characterized. People know what are the residues that are involved, what protein kinases are involved. But amphetamine efflux process is still not clear. Here's the model that most people consider, maybe it happens. It's true. So here you have amphetamine comes in and sodium chloride comes in, right? So you get this gradient. Then amphetamine, it doesn't show that. Amphetamine gets into the vesicles and decrease the dopamine in the cytosol, so you have accumulation of the dopamine, and also cause the signal transduction, which is induce the calcium efflux here. So what people are hypothesizing that is that the efflux process occurs due to a phosphorylation of the residues that are either on the A-terminal or C-terminal, or any residue that are on the intracellular loop. So what they were saying is that when the residues are phosphorylated, the protein, the transporter, undergoes a conformational change. So at the beginning, this is called outward-facing phase. So the transporter welcomes the ligand binding, the dopamine transporter. But once the dopamine comes in, amphetamine comes in, the protein is phosphorylated on some certain residues, then the outward gate is closed, the inward gate is open, so you get a dopamine, allow dopamine to bind to somewhere here, it can be transported outside. So this theory has been tested in the past 5 to 10 years, and there's no consensus on what residues are really involved and what, like, is there a really conformational change for dopamine transporter? But we do know that certain residues are involved in amphetamine-induced efflux, and they alter, they are responsible for the intracellular dopamine binding affinity for dopamine transporter. So what they did is they mutated certain residues that are on either C-terminal and N-terminal and the intracellular loop, and look at whether it alters amphetamine-induced efflux. So this is just a hypothesized model. I don't require you to study much, but just understand a little bit. This is hot topic. Everyone is working on it, but there's no consensus on this. Okay, so again, just to summarize the amphetamine, so it works on the dopamine transporter. It also works on the vesicular monoamine transporter. It also has effect on this the enzyme, the degradation enzyme. So, but keep in mind is that amphetamine works differently from cocaine. Amphetamine causes dopamine increases through efflux. Cocaine increases dopamine through blockade of dopamine uptake. Dopamine transporter is also subject to internalization, just like a GPCR. So you probably already know some GPCR undergoes rapid internalization when the ligand is bound. Like a beta-adrenergic receptor, it undergoes internalization within seconds when the ligand is bound. Dopamine transporter, it takes a little bit longer time. It's not a rapid onset internalization. But dopamine transporter, it undergoes a rapid internalization and recycling. So here is a tracking of vesicle movement. This does not necessarily mean, this is called a TERV. It's a fancy advanced microscope tracing of vesicles. So this is a work, when I was a postdoc, my lab was doing this. So I really like this work, so I want to show you here. So when you give, this is neuroblastoma into a cell, and which stably express dopamine transporter. So if you give dopamine, or the control, which is buffer, and then you start to monitor this vesicle movement towards the membrane, and you can see that this is a baseline. This is 0 seconds before the drug was added, and you can see 60 seconds. You can see the massive, this yellow thing towards the membrane, suggesting that the dopamine transporter containing vesicles are moving towards the membrane when the ligand is on board. And here, 120 seconds, which is 2 minutes, you can see this is even more trafficking a vehicle treatment, you see the steady movement. And here with the drug, the dopamine, which is a substrate of the dopamine transporter, you can see rapid trafficking. Okay? Same thing with amphetamine. This is 60 seconds and 20 seconds. This is baseline here. So you can see when you put amphetamine in the culture medium, you see the trafficking of the vesicles moving towards the membrane. You see more here. So like the trafficking occurs within a minute when the ligand is on board. So dopamine transporter undergoes a rapid internalization or recycling process. Yes.
What does AF stand for?
This is a quantification of under the curve, the area under the curve. So here shows a dose-dependent increase of the transporter trafficking. So I just showed you here within the 2 minutes, you see the increase of the dopamine transporter to the surface, right? If you do 30 minutes, what would happen? 15 minutes, 30 minutes, an hour. Tyler, what would happen? The recycle back, and what would you observe on the surface? They're recycling to the surface. What happened after if you do longer exposure? So when you do the GPCR, you guys talk about GPCR, right? So when you give ligand, you see internalization right away. If you do longer exposure, what happens?
So become desensitized.
Correct. So the same thing with the dopamine transporter. So you see the rapid recruiting of the dopamine transporter at the early stage. When you do longer exposure, they are going to internalize. Instead of recruiting to the surface, they are going to be internalized. And it will take much longer time for the transporter to recycle back to the surface. So if you do the ligand binding assay at this point, if you do the time-dependent ligand binding assay, on the surface dopamine transporter, you would see increase at the beginning, then no change, then decrease. And it will take much longer, a couple hours, for the receptor to go back to the surface. So what causes the internalization and recycling of the dopamine transporter? There are a lot of experiments, a lot of people have done research in this area. So I'm going to summarize this here. So for the dopamine transporter to be rapidly recruited to the surface, it requires a protein kinase called PKC beta. If you knock out PKC beta, you won't see the rapid recruitment of the dopamine transporter to the surface when you give the substrate. Okay? Another kinase that is involved in the internalization of the dopamine transporter is called CAM kinase 2. So if you knock out the CAM kinase 2, or if you overexpress a dominant negative CAM kinase 2, you won't see dopamine transporter internalization. Okay? So these are the two protein kinases that are involved in dopamine transporter trafficking and also dopamine transporter function. Okay, here are the RAP proteins that are involved in the dopamine transporter. The RE means the recycling endosome, early endosome. So when the transporter recycles, it goes to the early endosome for internalization, then goes to the recycling endosome for the receptor to go back to normal. Okay? So here are the detailed structure of dopamine transporter. The one point I want to make is that based on the literature so far, it looks like dopamine transporter phosphorylation is the key for dopamine transporter function and the trafficking. So if you look at all the residues here, there are serine threading residues. They are all substrate for protein kinase phosphorylation, right? So you can see how many in here that can be. This is a PKA, which is the protein kinase A binding motif here. Here's a PKC, protein kinase C binding motif here, and here is a CAM kinase 2 binding motif here, and somewhere here there's a PKG binding motif here. Okay, so there's a glycosylation site here. There's also phosphorylation site. So you can see throughout this structure, the residue here, there are so many sites that can be phosphorylated by different protein kinase. And some residues has already been mapped out, like the residues here are key for the dopamine uptake, and there are residues here and residues here are key for dopamine efflux. And some of the residue here, if you mutate it, it's going to alter the dopamine transporter trafficking. So the take-home message is the dopamine transporter is a substrate for many types of the protein kinases, and which leads to alteration of dopamine transporter function and trafficking. Okay, again, if you give prolonged exposure, if you give the dopamine transporter prolonged exposure to the agonist, it's going to eventually cause a downregulation, right? So there's this one elegant study, and I really like, is they found out the dopamine transporter are located in different compartments of the membrane. At the beginning, we talked about cholesterol is interspersed across the plasma membrane, right? So it turns out the dopamine transporter, there are two different populations of dopamine transporter location. So one population is located in the non-wrapped, means this region does not have cholesterol, okay? And there's membrane wrapped region is cholesterol, there's a tons of cholesterol in there. So you have dopamine transporter located in the cholesterol enriched region and also non-enriched region. So for the transporter located here, if you give prolonged exposure to the agonist, eventually they are going to undergo internalization through cholesterol-encoded pits. Okay, so basically they are going to internalize and pinched off from the plasma membrane. And for the transporters that are located in the cholesterol-rich region, they are going to be internalized through caveoli-dependent signaling pathway. Okay, so they undergo different signaling pathway. But one protein kinase that is responsible for both for both pathway of the dopamine transporter downregulator, which is protein kinase C. Okay, here just give you an example of how to look at the downregulation of the dopamine transporter. And there are many things that are involved in here. So ubiquitination, phosphorylation, palmitylation, they are all post-transition modification, and also as long as also like glycosylation, in the same category. Flotilin is a protein that is affiliated with Tyler liver wraps, correct. There's some internalization motive. That means there's a sequence that needs to be there for the transporter to be internalized. So as I said earlier, dopamine transporter does not act alone. There is a whole network that for dopamine transporter to interact with. So, so here's the dopamine transporter, and here's the dopamine D2 receptor. They form heterodimers. If you delete one, you compromise the activity of the other. So there's one elegant study looking at the D2 receptor dopamine transporter dimers. They found the dopamine D2 receptor, the third intracellular loop of dopamine D2 receptor binds to the N-terminal of dopamine transporter. So they designed a peptide that interrupts this binding interaction. And what they found is if you disrupt the binding, the dopamine D2 receptor activity is reduced, the dopamine transporter activity is reduced. Okay, so dopamine transporter does not act alone. And also we have syntaxin 1A, which is known to bind to the N-terminal of the dopamine transporter. And what this does is it regulates dopamine efflux. Okay, so if you knock down syntaxin 1A, you will see less dopamine efflux. Okay, here's a flotilin we just talked about. Here, if you delete this, it causes dopamine transporter. The dopamine transporter does not properly position on the plasma membrane. And here are the ubiquitination site for the dopamine transporter. Does anybody know what ubiquitination does? degradation pathway. Second, it's impede dopamine transporter trafficking. Okay, so you would have altered, delayed, or no internalization or no recycling. Okay, so a long time ago, everybody thought ubiquitination is responsible for the degradation pathway. It's not the case anymore. And you have this SH3 binding domain that combines a lot of like a PTZ containing molecules. Then you have this ring novel molecule that binds to, that is responsible for the dopamine uptake. And you have this chem kinase 2 that binds to the C terminal. Then you have this alpha C nuclei that is associated with the Parkinson's disease. So if you knock this down, you would have less expression of dopamine transporter. Okay, so and here's a PARC gene that is also, the PARC protein that is also associated with the C terminal of dopamine transporter. So as I said, dopamine transporter doesn't work alone. So they work in partner with a lot of proteins. So the third and the NET share a lot of sequence homology with the dopamine transporter, but I want to make one point is that here are all the threading and serine residues that can be phosphorylated by all the protein kinases, okay? Just like dopamine transporter. You see how many residues that can be phosphorylated by PK, PKC, PKG, chem kinase 2. And serotonin transporter undergoes undergoes the trafficking regulation by the protein kinase also. So you do see some similarity. You know, there's a large extracellular loop, intracellular N-terminal, intracellular C-terminal, and this is also same thing as the dopamine transporter. This is a binding site for a lot of proteins like receptors, like protein kinases, and also some fusion proteins like syntaxin is also binding here. So norepinephrine transporter, same thing. You see more residues that can be phosphorylated by protein kinase. Based on the literature, it looks like when you mutate, most of the residues, the phosphorylation residues here, it does not alter too much of the uptake function. But a lot of residues here, like the N-terminal or C-terminal, when you mutated them, it alters the efflux function of the transporter. Okay? I'm not going to talk in detail about the third and NAD, but I know that the two students who assigned the paper who's going to, one of the papers on CERT and chem kinase 2 you're going to do. And another paper is one of the DNA polymorphism you're going to, and talk about, this paper is looking at what does this polymorphism do to the dopamine transporter function and trafficking, correct? And at the behavior level, what does it do to alter the behavior response? If somebody wants to learn more, here's a paper talks about norepinephrine transporter and syntaxin. This is an excellent paper, and from Randy Blakesley, who's devoted his time, his research into norepinephrine transporter. So if everybody wants to know more, this is a paper to read. Okay. Let's talk about VMAT. Vesicular monoamine transporter also has a 12 transmembrane domain. Has intracellular N-terminal, has intracellular C-terminal. The difference between the VMAT and the monoamine transporter is it has a large extracellular loop. The first extracellular loop is very large. For the monoamine transporter, the second extracellular loop is large. Okay. There's VMAT1, there's also VMAT2. So VMAT1 is primarily in the peripheral nervous system. VMAT2 is very abundant in the brain. For most monoamine vesicles, the VMAT2 is the predominant form. again, here, you know, when you have the substrate that is transported into the vesicles through the VMAT, the protons is going to dissipate it. So there's a variation of the VMAT here. It's the proton, how many protons that get dissipated. So you guys can just focus on the VMAT. I'm not going to put anything on here because you're going to learn it later on. The cholinergic vesicle is the most well characterized vesicle so far, and you can see this diagram. So many proteins have been identified on the vesicle membrane, and what I want to point out is that here is the proton pump, the ATP is pump, and here is the VATPS pump to pump the proton in, and here's the vesicular monoamine transporter, vesicular acetylcholine transporter. You can see in and proton out. So what I want to mention a few things here is that there are a few proteins, synaptogyrin here, synaptophysin here. So these are the proteins that are key for the vesicles to be fused to the plasma membrane. So when the stimulus comes, the vesicles dock to the plasma membrane and fuse to it, then the vesicle undergoes exocytosis to dissipate the dopamine or acetylcholine into the synapse, right? So if you mutate any residues of this protein, you're going to see a less effect of the fusion of the vesicle to the plasma membrane. Okay, so these three proteins are the key for the vesicles to dock on the plasma membrane. There are a lot of publication on that. Okay, so lastly, I'm going to just touch a little bit on the effect of amphetamine on vesicular monoamine transporter. I already talked about that, but I want to say this one more time. Okay, under the normal condition, you have, dopamine, let's say, get transported through the plasma membrane into the cytosol and get packaged into the vesicles through the VMAT. Okay, so if the stimulus comes, this vesicle fused to the plasma membrane and it goes endocytosis. For the amphetamine, what it does is it gets transported into the cytosol, and the amphetamine acts as a false neurotransmitter, like a dopamine, and get transported into the vesicle and depletes dopamine out. So you have less dopamine. So chronic amphetamine exposure eventually will cause depletion of the dopamine in the brain. Okay? But they can get recovered to some degree. Okay, so what do we know? What's the association between dysfunctional dopamine transporter and the diseases? Here, I just want to give you a few examples here. So there are no dopamine transporter mutations or polymorphism that are associated with monoamine transporter, and the mutation of the residue of certain residue can lead to is associated with the behavior phenotype of a lot of disease. So here we have dopamine transporter mutation A50. This 559 means the amino acid residue on the dopamine transporter. There's a mutation from A to V, and what it does is it reduces dopamine efflux and associated with ADHD symptoms. Okay, there are two other mutations. What these two mutations do is that cause the reduction of dopamine receptor protein level and infantile Parkinsonism and dystonia symptoms. Okay. For the third, there are many polymorphism, and they are associated with a broader range spectrum of diseases, such these two SNPs are associated with anxiety, bipolar disorder, OCD, autism, and here here, this one is associated with OCD, and this is associated with autism. So NET mutation, this mutation causes decrease of NET level and depression. And VMAT, there are alcohol dependence and Parkinson's disease. So by studying each residue, we can understand what does this residue do to the receptor, to the transporter expression, to the transporter function, either the uptake and efflux, and understand and establish a direct association between, residues on dopamine transporters or NET or SERT with the disease. So how do we study dopamine transporter, and how do we study, what are the approaches we can use to study this? So if I give you a question, I said, please design experiment and test that this molecule, this proteins may be involved in dopamine transporter function or trafficking. So what are the questions you should ask? So first, the transporter function says a uptake and efflux, right? So you need design experiment to test, how can you manipulate this protein and test whether it has an effect on the transporter reuptake and efflux function, right? And also, does it alter the iron flow? Because that's the key for the transporter to work, right? For the transport of the substrate, because it maintains the electrochemical and the neurochemical gradient. Does it alter the surface transporter expression, or does it alter the internalization rate or recycling rate, and the compartmentalization of the transporter, like cholesterol enriched compartment, cholesterol not enriched compartment. Again, when ligand binds to the transporter, it causes signal transduction, mostly mediated by phosphorylation of the protein kinase, right? So you would expect to look at the downstream signaling of the dopamine transporter. So the approaches that are commonly used in the literature are gene knockdown, use siRNA, gene knockout, use the CRISPR-9, overexpression. If the cell does not have much protein to start with, that's a good approach. And also, there are a lot of research on manipulation of the residues. If you think they're involved in protein kinase phosphorylation, like PKC, you can, if you know, let's say, this residue is involved in the PKC phosphorylation, and this when you mutate this residue, it's going to abolish the PKC downstream signaling pathway. So what you could do is to see if the novel protein modulates the site, you can mutate the site and see whether it modulates PKC phosphorylation of the transporter, right? So that's a common approach in the transporter research. And also, as I said, transporter doesn't work alone. It also binds to other receptors of protein kinase as a dimer or tetramer to work. So you can design peptide to disrupt this protein-protein interaction, or you can just mutate a few residue and abolish this protein-protein interaction to see what does it do to the transporter function, correct? Internalization and recycling both require protein kinase. You could mutate the residues and see what it does to the desensitization and resensitization of the transporter. Okay, so these are the approaches and questions people usually ask when they study transporters. I think I'm done here. Any questions? Yes.
This is a little off track, but just out of curiosity for methamphetamine, how does the mechanism alter between that and amphetamine that it causes toxicity to the cells?
Methamphetamine is a substrate, so it's the same as amphetamine. But methamphetamine causes the toxicity has nothing to do with the dopamine transporter. What it does is it stimulates the oxidative stress pathway, so you get more dopamine once it's released from the vesicle because the inflammation process, so more dopamine is going to be degraded. It has nothing to do with